Thanks for inviting me to present today, and this is really exciting. I have a lot to cover, which we did in the last couple of years with CDeep3M, so I'm going to jump right into it. As Luciano mentioned correctly, I'm now in Berlin. Before that, I used to work in Mark Ellisman's lab at UCSD. But since June last year, I'm setting up my lab here at the Charite in Berlin. I can also turn on my video. Can you see me now?
Yeah, perfect. Yeah, we can see you. Yeah, nice to see you. Very good.
So yeah, CDeep3M is sort of a plug-and-play platform, I would say, for large-scale deep learning-based image analysis. And okay, so this was born really out of a need for analyzing our what we care about, which is volumetric electron microscopy data. And we want to analyze brain structure with that. So as you can see, we have different types of volumetric EM data now, like this electron tomography on the left that gives you some really high resolution images. You can see mitochondria, you can see ER. You see on the lower end the nuclear envelope and the DNA inside the nucleus. On the right-hand side, you can see a serial block phase EM dataset at a sort of smaller view where you can really follow Golgi apparatus and mitochondria very well. And yes, many of you might already know this serial block phase EM data can generate extremely large data. And just to give you sort of a overview, if you're not familiar with this, so many in the field are doing this connectomic types of study where large-scale images of a brain tissue are taken, and we will then reconstruct the neurons, where they connect, how they connect, but also intracellular features. And when I speak about large scale, so typically that means that a data set can be several terabytes, but now it's starting to reach petabytes in not only the Allen lab, but so this is something to bear in mind when we develop for future projects that this extremely large scale is gonna come. So in here you see now mitochondria and the nucleus in the middle, and all of that is being reconstructed from EM dataset. So these microscopes can acquire hundreds of gigabytes a day, and they can keep running for several months. So the scale is becoming bigger and bigger. At the same time, we still have the same high resolution. So all of this is being done without sacrificing resolution. So here you can see the reconstruction of ER and mitochondria from a single Purkinje cell in the cerebellum of the brain. And you see a very elaborate mesh of endoplasmic reticulum, and you saw also a lot of mitochondria appearing. So that's kind of our targets that we are interested in: reconstructing circuits, reconstructing intracellular organelles, and all of that in the brain, where actually the organization is extremely complex. And as many before me obviously already noticed, is that doing this is really a machine learning task. Humanly not possible anymore, and we can build on developments in the industry that segment images, for example, for automobile or for other purposes. But there is a lot of sort of added challenges. So because we are dealing with biological specimens, these are these are very large images. So this is not very typical in the industry that here we're dealing with multi gigabytes per image on a data set. This means multi-terabytes to petabytes, depending on how sort of what kind of acquisition you're using. And the images are at the same time quite noisy. As you maybe see here, there was a Gaussian blur applied because typically you start to see a kind of noise from the microscope. And something that's extremely challenging is that we're dealing with a fairly small training data sets. So unlike Google or Facebook that have access to millions of faces or cat images or whatnot, we're really dealing with humanly annotated training data, and that's a labor-intense process. So we are typically restricted in that matter. And when we look at the structures, you can maybe realize that, especially in these EM data sets, different structures can look somehow, similar, right? This is a black and white image, and the membranes of this cell, they're only contrasted by darkness, and the same for the ER in the center of this dendrite going up here. And this makes it, I think, a lot more challenging as well. We have also fluctuations through the imaging process in the intensity levels, and there's nothing that we would call a typical acquisition parameter, because staining intensities can vary, and especially, you acquire a data set one day and the next day you will not be able to acquire exactly the same conditions. So we have to kind of account for this, and that's one of the bigger issues in deep learning, is that you train a model and then it doesn't generalize very well, and it would not work as well on a different data set. So this is something we have been working hard on addressing with CDeep3M as well. And then there's additional sort of issues like misalignments across C, for example. So sort of the starting point before we started with this project was that there was many image segmentation challenges, and people would start to propose architectures and workflows. But then when you would try and apply it on your data set, you would still need to, spend a decent amount of time to actually implement that yourself. So you would need dedicated software developers if you were to start from scratch and reproduce something. And obviously we wanted to do that at the large scale. So the sort of goal of CDeep3M was that you would need no programming knowledge to use AI, and that you would not even need the hardware. You wouldn't need to make that huge investment in the beginning to buy GPUs that do the heavy lifting. And that's what is led to this publication in the Nature Methods in 2018, where we used cloud-based deep learning. So CDeep3M originally stood for CE for the cloud. So we used AWS, the Amazon service, for that in the beginning. And we sort of tried to really facilitate this and break it down into the two essential steps, which are training and prediction of the network. At the same time, we did a lot of development also to do the tiling. So obviously, if you deal with a terabyte data set, you cannot load it into a memory, so you're breaking it apart. You're processing the tiles individually. So that's what we would call the pre-processing steps. Then you would perform the prediction, and then the post-processing steps. So all of that is neatly integrated into CDeep3M, so you don't have to worry about it. At the same time, there's often an edge artifact when you apply a deep neural network and you perform a prediction, that the performance is much better in the center of the image than it is towards the edges, where the network cannot really decide anymore if your prediction is as accurate as you would expect it to be. So we built in this overlap between neighboring tiles, and then everything gets merged in the end automatically, and the user just gets the final product without having to worry about these steps. So what that allowed us at the same time is that because we're dealing with smaller tiles, and we can break apart the processes that are happening in the background. And if I sort of remind you, the prediction part is what really needs a GPU. So the pre-processing part, where we're extracting sub-areas, does not need a GPU. We're performing also image augmentations at this step. Then we're using the GPU to do the predictions. And in the post-processing steps and the ensembling, we also don't need GPUs. We are only using CPUs. So we implemented what we call a producer-consumer scheme, where basically individual tiles get pre-processed by the CPUs, and then this first package gets passed on to the GPU, and then the last step, it gets processed by CPUs again. And in parallel, while the GPU is running package 1, package 2 is pre-processed by the CPUs. And this sort of really comes down to how do we utilize our resources as efficient as possible. You don't want to sit on hardware that costs tens or hundreds of thousands and not utilize it ideally. And that was sort of the first essential step in this process. And so we obviously then went on and started to apply this to large data sets. So what you see here is sort of a 10,000 by 14,000 by 400 pixel wide data set. So I would call it medium size data set. It's not the largest we're processing, obviously, but it's reasonably, to show that the pipeline works. So this was also 2018 still. So, and we trained the network to recognize vesicles, and you can see, even though from distance they seem very easy to distinguish, when you look much closer, you can see that it's actually not such an easy process. Also, the vesicles are three-dimensional objects, and depending on your cutting thickness, through the specimen, you actually can see only one plane of the vesicle or 2 or 3 planes. So all of these things are important to take into account when you're processing large data. And for verification, we have one area where we do the training, and then we do the verification of the performance on different areas, or validation in that sense. The CDeep3M obviously does not only work for EM data. So if you're interested in fluorescence microscopy or micro-CT, we also used it for calcium imaging data. You can basically segment different kinds of images and train your network specifically for your own data set. And in terms of validation, already back then, we sort of were at the level of different human segmenters. So here's a comparison of the F1 value and the Cheka index, just comparing CDeep3M and different humans, their sort of human-to-human comparison. Interestingly, when we look at sort of at this data set and we look at where the errors are occurring that the network makes, a lot of it has to do with how humans label data versus how the computer labels data, and where the boundaries are being exactly set. So you see in sort of pink what CDeep3M only predicts, in green what the humans only predicted or labeled, and the overlap in between them is white. And you can see that there's kind of a halo around the mitochondria here. And the reason is that CDeep3M sort of predicted the mitochondria a little bit wider than the humans in this case, labeled them. So when we started to sort of account for this, and this has been also noted by other people in the field, that actually those are not essential errors, because the mitochondria just get a little blurry around, and the humans might say, okay, I'm labeling the interior of the mitochondria, versus the computer also labeled the membrane as well. And so we introduced this 1 or 2 voxel exclusion zone and remeasured the sort of performance of the network, and you can see if you start to remove these areas in the calculation, and in this case, for example, the computer just labeled a mitochondria in the 3rd dimension where the humans already stopped. So that's why this starts to appear right in here. So, and then our performance goes even further up and exceeds the one of human sort of agreement. Already back then, we also invested in enabling transfer learning, which can save you time and money. So in this case, we had a pre-trained network. You would apply it for a different data set, and you see a lot of blur coming out. And after retraining it with a small amount of data set, on the lower right corner, you can see all of a sudden we do get crisp membrane predictions again of the same cell. And whereas if you would retrain with the wrong training data, you wouldn't get that kind of improvement. So in this case, the training time was reduced to about 1/10, and the training data about 1/20 of the original training data. And why do we care so much about membranes? It's simply in that field of connectomics, how most people go from the EM dataset to predicting the 3D structure of the neurons. So once you predicted correctly the membranes, it's much easier to actually agglomerate the interior regions and decide that on the upper middle you have a presynaptic site with those vesicles inside, and then in the lower middle you see the postsynaptic spine. After that, of course, for us, this was not the end of the story, and still is ongoing. So, as I mentioned before, when you go from one data set to another, you often see a drop in performance because this was not seen by the network before. So we really are working a lot on generalizability of the models. So we want broadly tuned models. So we are training them with multiple training data sets, ideally coming from different image volumes. So we implemented that in CDeep3M to make it easy for users to also use that functionality. We also integrated additional augmentation strategies that are widely used in the field. We divide them into primary, secondary, and tertiary. Primary being rotations, flips, inverts. So basically everything that does not change the image is a primary augmentation. Whereas secondary are these typical noise addition, denoising, sharpening, blurring, contrast modifications. And tertiary are resizing operations. And there's a reason for dividing it into those three, because when you train your network, if you only do primary augmentations, you're not changing your image. Whereas if you do secondary, you are actually training the network to also recognize blurry images or sort of, yeah, altered images. And with the tertiary, you're actually training the network to recognize images at a different, or objects of a different size. And so this can be problematic if you're doing too much of a resizing operation. Your vesicle might all of a sudden, you know, look 5 times bigger and look a lot more like a, you know, different object in the cell. So we are sort of not always using secondary and tertiary augmentations, but they are enabled, and we do use them when we want to train a model very broadly. We also implemented automatic image enhancement. So on the right side, you see this scrolling through, and the first image is the original, and then comes the enhancement. And this is fully built into CDeep3M now. We also improved speeds, the speed of the overall prediction performance. We built in automatic overlays, and everything is fully backwards compatible. So if you train the network in the CDeep3M1, it still works with CDeep3M2. And so now I want to basically jump from development to deployment. How do we get the sort of platform to the users? And I spoke about that we used the cloud originally, but obviously there are many different ways to run a deep neural network. You can run it locally, you can use supercomputer clusters, or your institutional cluster. You can use cloud computing, as we sort of pioneered with the CDeep3M, and then you can also use edge computing, which is sort of a variant of the supercomputer clusters, but often with newer infrastructure. So for local, the only advantage that comes to mind for me is that it's the most familiar for people. But it's for funding, it's extremely expensive to actually have these high-end hardware sitting around in different labs. Most of the times it's actually underused unless you're using schedulers, and very often you have compatibility issues with a particular software, so you might need different systems for Mac, Windows, and the hardware in this field really devalues over time very quickly. As some of you might have noticed, NVIDIA is putting out new GPUs every half a year by now, or a year. And sometimes the price can drop like by 50%, and your old hardware is outdated in 2 years or so. So I'm sort of advocating for using alternatives like clusters or cloud computing, but we do enable also the local installations. So the clusters really have the advantage. You don't have bottlenecks, because the jobs just get scheduled when there's free hardware. You have extremely high performance, and all of this counts for cloud computing and edge computing at the same time. Some of the difficulties for users is sometimes, okay, how do I get my data in and out? And particular for the supercomputer clusters or institutional clusters, you're limited what you're allowed to do, and sometimes you're dealing with some outdated distributions because they have to guarantee some long-term maintenance. But here, edge computing comes really in as something that has a lot more recent sort of distributions, and is becoming more and more available. Cloud computing for me has a sort of special place because it's a lot more convenient for developers and end users, because you really know what the end user is using, and you basically have a very closed environment and that you can develop on. And just to give you an example, so this is the cost in 2018. So a sort of extremely high performance V100 GPU with this expensive hardware would cost you about $3 an hour, which the system probably price range would be $20,000 to $30,000 if you would buy it. So if you pay $3 an hour, you get about 6,600 hours worth out of it. If you say you're using it 24/7, this gives you more than half a year of runtime on this system. And similar on the sort of P3 16x large, which was the largest nodes at that point. Now, what happened in 2019 is that NVIDIA brought out new GPUs. This propagated to AWS. The prices dropped significantly. So within 2 years, the price of use is about a sixth of what it was 2 years ago. So you can get a very decent GPU for 50 cents an hour. So if you would invest that money into a workstation, you would probably pay about 5,000 euros. And that would give you a runtime of 566 days if you would use it 24/7, which unless you're using a scheduler, you will really not manage. So you see, I'm maybe a little bit biased in towards cloud computing. I think once funding organizations realize this, they will stop funding labs for buying hardware, and they will make large investments into cloud or institutional clusters, and that will sort of give everyone a big advantage. But since we're aware that people do still like to own their own hardware for some reason, we also do enable this. So CDeep3M is distributed in a way, so there's the base code which lives openly on GitHub. We push that in, we build a Docker container out of it, and then this is basically containerized. You can use the Docker container on your local workstations by just installing Docker. You can use it on a cluster or remotely very easily. And at the same time, these containerizations for us propagate into a singularity container. They also propagate into a CloudFormation template. And what I will hopefully still have time then to demo the preview function, which runs on a decentralized cluster. It does not directly propagate. Actually, it's the source code that propagates into the cloud installation on Google Collab. And that's sort of a finicky thing of working with Collab. So this is only something we're doing on the side. So very briefly, and I will demo this in a second. So basically, you can either upload your own images through the web browser or browse through very large data sets that we have hosted on the cell image library. You can then test out different trained models and try out different settings for CDeep3M. You click submit, and then basically this in the back end sends a job out to our cluster, which runs that job for you for free, and you receive an email, and after a couple of minutes, depending on the settings, and then you can inspect your results. So you can actually use that to process your own data. It's a bit limited in terms of volume, because obviously we cannot have people segment terabyte-sized data online, but we provide then easy instructions. So my ideal use case scenario is you crop out an image from your large data set and you want to test out a model. And you want to know, does that model really work on my data set? I'm going to just submit it here, and before investing any thought other than that, you try out the model and all the settings. And if that's satisfactory, you can either choose to run it on AWS locally or on a supercomputer cluster. If it's kind of there, but you think it could be better, then you can still generate more training data, and you can also run training on our platform online. So the reason why we're basically, you just need to request an account in the beginning that's for free, and you will not be charged anything ever. We also don't use any of the data that's being processed on there. So the only reason why we're requesting you to create an account is that we don't want inappropriate users using it, and you might want to actually share the results. So you're receiving a link for your results once the process is finished, and that also means you don't have to sit and wait. You can disconnect. And you come back later, no matter how long the job runs. So I mentioned we also do have a Collab implementation, but yeah, as I mentioned, it's kind of restricted in what you can do with Collab, just because it's not a commercial provider, and so it's not Google Cloud, it's this free product, so you're very limited in the data. The interface is more involved. You need to install your dependencies. I mean, we do that, basically we provide that for you, but you need to wait every time for the full installation to finish, because they don't allow AMIs, like AWS, for example. And Google disconnects you when you're inactive. So on the website, you receive an email, you don't need to wait, whereas here, you actually do need to wait for your process to finish. At the same time, because we are on the website now, we really have the possibility and implemented to connect this to a model. So this is our online connection of pre-trained models. Each of them has its own DOI, and I can show you then. So you get credit for uploading your model if you choose to do so. And just in sort of example application here, there was no transfer learning applied yet. This is a completely unseen data set, so you see some of the models. So this sort of violet color is where predicted synaptic clefts. We didn't train for it, so there are a lot of false positives in here. This is just to show you, so the vesicle predictions are pretty good. The mitochondria predictions are not bad at all, and the membrane predictions are also not bad. And for each of the models, you can basically just say, okay, this is the DOI of the model I used for performing this segmentation. So this is extremely important for us in terms of reproducibility. How do you share what you did? You can very simply keep track of it by using this DOI. So we're hoping that we're sort of pushing a little bit the standards in the industry. How do we share our progress in deep learning with others? So everybody can use deep learning for large-scale image analysis. You don't need to know how to program. We want to increase the reproducibility. Our code is online executable. It's fully linked to a database of images and also a database of trained models. So you have also the image browser, and everything is sort of working end to end. And we also want to facilitate in general the sharing of deep neural networks, so you can upload your own models, and you do get credit by assigning the DOIs. So there are many sort of friends and colleagues and collaborators now that already used CDeep3M in their work. So on the left side, you see this work from Tom Steinkellner in the NASCO lab together with Daniela Boassa. So here we did segmentations of the presynaptic vesicles, and we have a tag for a certain neurotransmitter, which is illustrated in color here, the vesicles that contain this neurotransmitter. On the upper right, you see microglia segmentation. On the lower right end, you see neuron segmentation with also vesicles and mitochondria and many of the intracellular organelles. And if I still have a couple of minutes, I'd quickly do a demo. Are we still on time?
Yeah, go ahead. Go ahead, Matt. It's okay. Certainly, I think everybody will be interested in seeing a demo. and very impressive platform. So covering a lot of applications. So certainly, yeah, go ahead. Yeah, actually, the AI Microscopy Symposium was initially meant to be pretty much just demos. An on-site event just with demos, but it turns out because of COVID, we had to turn to a kind of an online event. So now we still very much welcome demos. So go ahead, yeah.
Okay. So I hope you can see my screen.
Yeah,
okay.
We can.
Okay, so this is the sort of landing page when you come to our online platform. On the left-hand side, you see what's going on with training. On the right-hand side, you see what's going on with prediction. These are the rough categories. And it splits into either create training, retrain a model, or upload your own trained model. And then you can track what you have done in the past with your trained models or your training data. On the right-hand side is the predictions. So this is maybe the most interesting right now. I'm going to start on this side. So if you go on your own images, you basically can upload your own images. And I'm gonna, this basically just jumped. You still see my full screen, right? Is that correct?
Yeah, I still see the full screen. It looks like you have two screens. So right now we see your slide deck. And now the web browser.
Okay. Yeah, so what I'm gonna sort of do is I'm gonna drag and drop the image that I wanna segment. Oh, I need to exit the full screen somehow. It disappeared. Let me stop sharing and this keeps jumping back and forth between screens.
Yeah, now we see again your screen.
Okay. Okay, so I wanna.
Now we see your screen again, yeah.
Okay, so I wanna upload my images. So you go to your own images, and I'm gonna do some test case. Like if you see this, that actually looks like a very faint image because somebody has not adjusted the contrast yet. So when you do that, obviously, you need to pre-process the image. So all of that is being built into CDeep3M. So I'm just going to drag and drop a couple of images here, upload them, and you see the progress of this. And then it jumps to the next step. So here I'm going to select which trained model we're going to use. And I'm going to basically choose one of those models. So it's either synapse detection, mitochondria, membranes. So there's a bunch of different models that we have now available. I'm going to use a SBEM and TM membranes. And then AUG speed is one of the settings. I might explain later how that exactly affects the results, but it affects the results and the speed. And then the depth of the neural network, either it's a 2D network that only sees one frame, or it's a 3D network that sees 3 or 5 frames. And then I'm going to click submit, and I'm going to receive an email in a couple of minutes. And this depends a little bit on how, what exactly your settings were. And okay. So it might take somewhere between 2, 5, or 10 minutes, depending on how large the image is and what your settings exactly are. So you basically get to this screen now, and it tells me my results will be here, but I'll also receive an email once this is done. So I want to basically jump ahead and show you what the results look like, because it would take, yeah, here you can see it took about 3 minutes when I did this. Or 2.5, yeah, 2 minutes, 15 seconds. And so what's happening in the background basically is it's spinning up a Kubernetes pod on our cluster. It's transferring the data, it's using the GPUs that are available there, and then it's returning the data once it finished processing. So you can see the image was now automatically enhanced from this sort of grayish image to really improve the contrast. You see the segmentations. So I've not chosen any particularly, image that we've trained on. This was a random image I sort of saw laying around, and then you see the overlay that's being automatically generated. Now from here, let's say this was, actually a crop from a really large data set, and you see, okay, I'm kind of curious, I want to apply this on large data. So you then go and you look here how you run it on Docker on your whole data set. So obviously you need to install Docker. It's just the containerization that's enabling all of this. Then you pull the Docker image and you run one command. You download the trained model. So you see the DOI is being used here to actually specify which model was used. And then you can go ahead and run the predictions. Same thing, you can use Singularity, which is used very often on high performance clusters, because it's a little bit more regulated than Docker, and you can use our Singularity file. You can also do the same on AWS. So for this, you only need to have an AWS account, click on Launch Stack, and the procedure is the same then. You also can try and do it on Collab. I don't particularly recommend it because, if you want to do a large data set, it's not going to, there are so many limits imposed by Google that it's not going to do what you want to do. If you only have a couple of gigabytes, that might still be a route forward. So this was basically functionality one, is that we want to do predictions on your own images. Now we also, I mentioned, we have the cell image library, which is a database that has terabytes of images from different projects that we have done in the past. And so there's one cerebellum, DLGN, and we can populate this more over time. You can also submit your images and say, okay, I'm done with the project. I'd like to share this with the community. Can you host it on CIL? And you can enable then doing predictions on there. So I'm going to click on the cerebellum data set, and you can see there are a couple of sort of pins here already, and then you can sort of scroll around and see the image itself, or the whole volume. On the right-hand side, you see, depending on your internet connection, you might need to wait a little, so I can scroll through there a little, and you can follow, mitochondria or whatnot. And then basically all of this is available for running image segmentation on it with CDeep3M. So you can just set a pin here and say, okay, I'm gonna do a segmentation here. You click on it and you do CDeep3M preview. And now you can choose which trained model again you want to use. You want to do memory and predictions on that. And let's say you want to use 3, 5, and 1FM, and then you click submit, and then it will tell you that it's processing the data. So first it's basically cropping that particular area out of the large image. It's spinning up a pod now on the Pacific Research Platform and processing your data in the background. So I'm expecting it would take maybe 5 minutes or so. So in what I can show you, I've done one of those before a couple of hours ago. Wherever you see a pin, you would get the previous results available. So in this case, there's one result on this particular pin. And then you can jump there and see the previous results. If there's more, you will see more results. And then you can look at the details, what was processed. There's basically track of the entire pipeline of how we do the processing. Here's the trained model that I had used there. In this case, I used AuxSpeed1, which is sort of the most time-consuming. And I used the 2D and the 3D models, and then it took about 8 minutes to process that image stack. So we can basically go through that. There's a little bit of a lag, but you can see how it now did predict. Since we didn't train on this data set, you can see there's a little bit of some prediction around the mitochondrion where you might think there's a, but basically that's, I would say, extremely good in terms of generalizability.
Oh yeah. Yeah, that's really good, Matthew. Matthias, I don't know if you want to have any final statements before we go to questions, but the demo looks great. Thank you for showing those two cases, how to use it. I guess if there's a lot of people using this platform, it will. Who is paying the bill? I was wondering.
Yeah, so this is NIH funded, and it's the Pacific Research Platform. It's expanding largely. So they want to go US wide and then worldwide. So basically, this is not a regular supercomputer cluster. This is edge computing. So this means you personally, let's say, are interested and you buy your own hardware. You can get it on as a node onto on the decentralized cluster, and you can run specifically on your nodes if you wish to do so.
Yeah.
So yeah, I think this is really an exciting route because for developers, you really have a lot of control. You don't get these random error messages where somebody just doesn't know how to install something, and like this, you really are fully aware of when an error happens, why it happens, and you can reproduce it easily. So. Yeah, very briefly, I would just want to acknowledge some of the people, not all of them. I'm not going to go through. So yeah, actually has been really critical recently to continue the development. Willy Wong is the web developer behind this, and Sean is a system administrator who has been also very important in the project. Daniela and Tom, who have been really important on this Synapse project that I briefly mentioned, and this is our ongoing collaboration. And I'm happy to take questions.
I have just one to get us going. So the first time I saw your work was probably 2018. I'm not really sure if there was a preprint before you had the Nature Methods paper. But I have a feeling I saw it a little bit before that. Perhaps I saw some of you presenting at conferences or something. And back then it was really revolutionary. There was nothing else like that. That would already at the time was AWS, so that would have deep learning pre-trained models that would do it in the cloud, and that would make it somewhat easy to do it. Like if you had an AWS account and you tinkered a little bit, you would get it to work. Can could you comment a little bit about how things have evolved in the last just 2 years? For yourself, it's clear. I mean, it's clear how much you've come along, you meaning the project. But more generally, what have you seen outside of the group that you were at that looks interesting and that kind of shows the direction that things are progressing to?
Yeah, so I think there are a couple of different sort of directions, I would say. So obviously, some people, rely on dedicated hardware individually, but when you look at sort of the connectomics field, where there was just recently a very interesting paper, and there was a preprint before by Sebastian Seong's group that they call chunk flow, and I think this is sort of all of these things are going to pave the way of how we do analysis when everybody is going to have access to these terabyte or petabyte data sets, and this is not a far future. This is a very near future. So it's going to expand. So how can we utilize resources more efficiently? How it's not the best idea to invest in buying your own dedicated hardware unless you're developing on it, because that was one of the points I really wanted to make about cloud or edge computing or supercomputer clusters. The other thing that's sort of happening at the same time is obviously more and more people are also really interested in how do we get everyone to be able to use deep learning, and I think that's a very important aspect also. So the presentations from last time, for example, using Google Collab, which for small data sets, it gets the tools into hands. Yeah. But yeah, I think there has to be a distinction between what can everybody do. But yeah, at the same time, I think more groups are now really trying to harvest as much training data as possible. And obviously this is improving the models. So that's happening on the side, I would say.
Yeah, that's kind of the race. And some groups have been doing that for so long, like Sebastian Seong and that whole initiative of citizen scientists, annotating by hand. When they started, I don't think anybody was even considering, like the general public thinking, I'm talking the scientific community, wasn't at the time thinking, okay, this is totally a machine learning thing. They knew, I mean, they are in that field, so they knew, but a lot of neuroscientists, even at the time I spoke with, just thought, oh, this is an interesting kind of fun online thing. But the amount of data that they've accumulated over, I don't know, it might be close to a decade now. It is for sure tremendous, and they have people from all over the world doing it.
I would say it's probably the most challenging of all tasks, right? The reconstructing a neuron over the full run length of an axon or a dendrite. This is a lot of errors that can propagate in that space. Okay, good. Let me double check if there's any other questions typed in here.
Yeah, this is a question. Yeah, does Matthias have any best practice techniques for sorting and accessing large data, especially petabyte-sized ones?
Sorting and accessing.
Do you have a? I mean, you don't use Omero, I'm guessing. Use something else, or maybe you do use Omero. Or no, I don't know.
No, I mean, I have to say that, so petabyte is I'm saying this is around the corner, but it's obviously this is not a day-to-day business. But we have internally at NICMER our which is similar to the cell-centered database. So this cell image library So this sorts our data for us. Everything is organized into projects automatically. When it comes off the microscope, it's synchronized and organized, so we don't have to do that by hand. And so this has been obviously been going for probably 10 or 15 years. And then if I want to synchronize it and make it public, I basically would just push it, from our local repository into the public repository.
Okay.
Yeah. So it's an internal solution. Yeah.
Yeah. That tends to be the case also in other places. So the CLI is another one which kind of expanded. I know it's used more broadly, but it initially started there, right, in San Diego.